Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Ingrid Holm, who's a professor of pediatrics at Harvard Medical School and is on the faculty of the Division of Genetics and Genomics and Division of Endocrinology at Boston Children's Hospital. Ingrid has been a pioneer at the intersection of genetics and pediatrics for pretty much her whole career, and we're going to do a deep dive today into some of the exciting things that are happening at this intersection and also what the future may hold. So Ingrid, thank you so much for taking the time, and I'm delighted to be here with you.
Yeah, but thank you for having me.
How did you get interested in this field in the first place? I'd love to hear a little bit about the backstory.
So I've been interested in genetics most of my life. My mother was a developmental pediatrician many years ago and did a lot of things in genetics. I've kind of known about genetics and pediatrics for a long time and very much interested in that area. And then as I've kind of gone through my career, there's been kind of some changes, which as time led to me to kind of start to be interested in really kind of implementation of genetics in pediatrics and really in the impact on families and really starting kind of in the research, obviously, but also then leading to research that has potentially would lead to clinical implications.
And one of the
genetics has been a big, I was going to say genetics has been a really changing field over the past 30 years.
Yes.
And so we went from very little genetic testing other than chromosomes in my career to now doing much deeper genetics. And that kind of evolution has led to kind of an evolution in my interest as those have developed.
Yeah, that is exactly one of the questions I was going to ask, and you, I think you teed it up really nicely. One of the projects that you've been involved relatively recently is BabySeq that many of our listeners might be familiar with. We had Robert Green actually on a previous episode of the podcast. For those who aren't, it's a pioneering genome sequencing program for newborns that's now, I think, on its second chapter. I'd love to hear a little bit about the origins behind BabySeq. When did the idea first come about? What was the research question or problem you were trying to solve? And then what did you learn through that first chapter of the study before we move on to chapter 2?
Yeah. So those are all great questions. So this really, this study kind of arose in part from a request for funding from the NIH to look at the implications of incorporating genomic sequence into newborn screening. And so that kind of led to us really solidifying our ideas. And a lot of it came actually from Robert's work in MedSeq. So Robert Green had done this study really looking at genomic sequencing in adults and incorporating that into clinical care in terms of risk. And so when this RFA came out, we were kind of here, children's kind of thinking about what we could respond to. And Robert came to us and said, why don't we try to do something that really builds on what had been done in MedSeq, but in the newborn screening arena. So that's really how the project itself kind of got going. But it really, it's one of these things where the NIH was just very interested in this question, which we also had become interested in. And then we've kind of built on that. So that was the first BabySeq project, was part of that RFA. And for us, the real goal is to really think out of the box in terms of newborn screening and think about really what is the future of newborn screening. And with our abilities and the cost going down for genomic sequencing and the real opportunities to screen children at birth for not just these critical metabolic conditions that can be, or hypothyroidism and these other conditions that are treated in that way, but can really start to look for other conditions that they would be at risk for later in life, in their childhood life. Things, cancer, cardiac disease, all sorts of things. We also kind of started to think about that that maybe everything doesn't need to necessarily be actionable. So a lot of families have children who have medical problems where they don't know what the reason for it is and go through this kind of diagnostic odyssey, and doing screening at birth and providing results back to families, even if there's no specific treatment for it, still would provide families with answers, prognosis, and kind of ending this idea of this diagnostic odyssey. So we kind of really started to think out of the box in terms of returning a broad range of results back to families that were of childhood onset, but not necessarily just actionable, but also things that would provide some kind of personal utility for families and their caregivers, and provide answers kind of in that realm. So those were kind of how we were thinking about BabySeq initially. That was really the first iteration BabySeq, which we enrolled families in the newborn nursery and in the NICU at Children's and at the Brigham. And we, I think very successful. We found 11% of our families, we found something to return in terms of monogenic findings. We found carrier status for almost everybody. We showed that the kind of psychological impact was not, there were no kind of harms being done in that arena, and the medical costs were not substantially kind of increased in this smaller cohort. The issue was that it was not a particularly diverse cohort. So we had approached all families at the Brigham newborn nursery, which is a very diverse nursery, and the Children's and Brigham NICUs are also very diverse in terms of kind of ancestry, socioeconomic status, that type of thing. But we just found that people who enrolled tended to be of higher socioeconomic status, white, non-Hispanic, because it was such a difficult time to enroll. So we're enrolling children in the right after birth, and most families are in the newborn nursery for a couple of days. And so that was kind of difficult to enroll. And so that we ended up with a certain demographic that enrolled. And so we realized that if we're really thinking about incorporating sequencing into newborn screening, we can't, it's not certainly not adequate to look at a European well-educated population. We really need to look more broadly. And so that led us to BabySeq 2, where the goal is to enroll populations that are underrepresented in research, and so primarily African American, Hispanic communities. And the way to do that is to go where the communities are. So instead of the newborn nursery, we're going to community health centers that have that serve these populations, and enrolling in those centers to really try to understand more broadly how would this, how do people of a variety of different, kind of populations feel about getting screening? What kind of results are we going to find? Are there any harms being done? So that's kind of the short history of the BabySeq project.
Yeah, amazing. And how big was the enrollment in the first BabySeq 1, and then what are you aiming for in BabySeq?
Yeah. So the first enrollment, so BabySeq 1 we ended up enrolling just over 350 babies. I should also just say this, note this is BabySeq is a randomized controlled trial of sequencing versus not sequencing. And I think that really sets BabySeq apart from most of the other studies that are being done kind of in this area. And the reason for an importance of doing a randomized controlled trial is that if you really want to understand the impact, we need to compare people who get it and people who don't.
Right.
And certainly in terms of psychosocial impact, but also in terms of healthcare, in terms of healthcare cost, medical outcomes, all of these type of things. If you don't have something to compare it to, it's hard to say, did you really make a difference? And so this is this randomized trial. And yes, I said a little over 350, and so a little over 150 were in the sequencing arm in BabySeq, in the first iteration of BabySeq. The second iteration, our goal is 500 babies, but we're certainly hoping to do, to be able to enroll more than that, and would love to be able to get up to 500, to 1,000 if we could, and are kind of working actually with some, to try to add some sites into the second iteration to increase the numbers. Because again, if we really want to see differences, the more that we can roll, the more differences that we see, and the more diagnoses we potentially would make, and potentially for rarer diseases if we enroll more.
Yeah, it is really different that it's an RCT actually, and allows you to do things that a lot of other studies would really struggle with, right? I'd love
Exactly.
to hear a little bit more about the measuring those psychosocial outcomes, I think you called them, because that is one of the major questions, I think, not just in newborn screening, but also predictive genetic testing or carrier testing for things later in life. And yeah, there's a lot of anecdotes on either side, right?
Yeah.
The potential harms, the potential benefits, but you have a unique opportunity to measure it very directly. How did you think about it in this case? What are you measuring? What kind of different axes are you measuring at the start, and then how long are you following up for in these cases?
Yeah. So all really great questions. So just first of all, to say that a lot of this is being, many of these psychosocial measures are being done at Baylor, which is a part of our studies at Baylor. And so Amy McGuire and Stacy Pereira at Baylor, the ones really who are kind of driving these studies of the psychosocial outcomes. So when BabySeq, the initial BabySeq started in the mid kind of 2015 or so, there was a lot of concern about psychological harms of returning risk information that wasn't completely certain. So we can't say 100% you're going to get this, but you're at increased risk. So there's a lot of concerns about that. And so a big piece of the initial BabySeq project, the big outcome was really this kind of psychological outcomes. And we're looking at things, a variety of different kind of domains. So certainly looking at parental depression and anxiety. We're looking at the relationship between the parents, the parental blame if you're returning some kind of results and it came from one side versus the other. Do these returning this information affect how parents view their children? There's something called the vulnerable child syndrome, and when you feel that your child is more likely to have a problem, are you protecting them in a way that kind of affects your relationship with them? So it's really, those were kind of the three big domains that we looked at. And what we found is that really we compared the control arm to the sequencing arm. And if we compared those who got a monogenic result back and those who didn't in the sequencing arm, among those kind of three groups, there really wasn't any difference. Everybody, first of all, were very below the cutoffs, in those groups. And there really wasn't an increase in any of these kind of negative outcomes in the initial BabySeq project. And so, and we're doing the same now and looking at the same domains in the BabySeq, the second iteration of BabySeq. We're certainly looking at that. And I think it's particularly important because now we're looking at kind of general populations that are, as I said, lower socioeconomic. We've kind of started to look at our demographics. Many of the participants are on Medicaid or CHIP, some kind of government-funded insurance. A lot of the families, the majority probably, are on WIC or receiving some kind of, infant help for infant formulas. The economic cut, they're certainly lower in terms of the household incomes. And so these are families that are more stressed in a way. They have a lot of other stressors. And are we contributing to that by then giving this information back? And so it's very important to look at all families in that sense. We did interviews in before we started the second iteration, we did interviews with mothers from the communities from which we were enrolling. And it was interesting because a lot of them, recognizing they have kind of a lot of these issues, feel that, well, if I had this information, at least I would have a little bit more control. At least it was something that I could have some kind of control over and understand. And so for a lot of them, it was very important to get this kind of information back for the sense of having a little bit kind of more control. So I don't know if I completely answered your question.
Absolutely. No, you did.
Yeah.
It's an area that I think is really interesting, and you've designed it in such a way that I think you have gotten really good data, but you're going to continue to and answer this pretty fundamental question of the do no harm principle, right? And making sure
exactly
that we understand that before focusing on the potential benefits, right?
Yeah, exactly. So we are trying to kind of do no harm. And I was thinking about, if you do this whole idea of doing a randomized controlled trial, if you do any kind of intervention, for example, in therapeutics, you do a randomized controlled trial. You do that first to be sure that it's getting what you want, and then you go out to other, have it be more general. So I think that's a good paradigm for this type of intervention.
Yeah, it definitely is. I was also curious about the breadth of results that you all made the decision around returning. So we've interviewed previously Wendy Chung, who's leads the Guardian project that I'm sure you're aware of. Also, Holly PA, who leads a newborn screening program out of North Carolina. And I'm working on the Genomics England group to do the UK one because I
Yeah,
think this is the future. But one of the things that's a little different between each program is the gene list. Almost everybody obviously returns the highly actionable set, but then the layers of the cake beyond that, whether it's potentially actionable because there are clinical trials or carrier status. I'm interested in how you thought about that because you've got not just the child to think about, but also the parents that there's potential for findings as well, and how much you constrain that or how broad you go is obviously a pretty big decision in these kind of programs.
yeah. So yeah, and again, that's a great question. I think that's part of what kind of distinguishes BabySeq from some of these other programs that are really focused on actionable, making diagnoses that are actionable. So we don't so much have a list. We kind of have criteria, and that has kind of led to a group of conditions. So we're not kind of starting up with the list and saying, what are we going to put on the list? We instead define the criteria as childhood onset, kind of a high degree of penetrance, and that there's either childhood onset or there's some action to be taken in childhood to kind of prevent disease that might be in adults. And so it's really focused on the childhood onset. And so the list is, there's probably, I think, 900 to 1,000 genes or conditions that are on that list. We have included a few kind of highly actionable adult onset only conditions based on the criteria from the American College of Medical Genetics and Genomics, kind of actionable, secondary findings to return in clinical testing. And so that listed from the ACMG is now, I think, around 80 genes or so. And so there are a few that are adult onset only. So the BRCA1 and 2, and I think at least one or two kind of colon cancer genes, I think Lynch syndrome. So whatever is on that list, everything on that list is on our movie return anyway. And then we've added those few actionable adult onset conditions. And that kind of came out of our experience in the initial BabySeq project, where turned out there were two families that had a BRCA variant and one that had one in Lynch syndrome. And we felt that because these are adult onset conditions that returned in clinical care in the secondary findings in babies, in anybody. So it's not we don't restrict in clinical care those conditions, and we felt that it was important if for families to know that they might be at risk. And so in our case, that in all those cases, one of the parents was also had that, and it impacted on their medical care. So again, that's, I think a different situation than a lot of the other studies. And again, the reason for returning things that may not be actionable is really this issue of personal utility, providing, ending kind of a diagnostic odyssey for a family that has a condition and they don't kind of know what it is. It provides families information when they're thinking about kind of reproductive, decisions that they're making. We also decided to return carrier status, so that is also a little bit different. Again, because it may be information that might be useful for families in terms of their reproduction. And we actually found in the initial BabySeq project that there was a baby who was a carrier for a condition that the parents then went to their obstetrician. They're having another baby, and they checked to see, because the baby had one variant and it was a recessive condition. And it turned out both parents had variants in that condition, in that gene. So the child inherited from one parent, but the other parent also had it. And so the parents, as it turned out, were at a 25% risk of having a child with that condition. So it actually, in that case, kind of impacted on, family care. So we are kind of, as I said, going beyond what a lot of other studies are doing, but we feel that's going to provide some really important information to add to the information that's being gathered, also in other studies about if you really push this out to really everything that you can find, what is the impact of that?
Yeah, how do you think about that as as a pediatrician, and how this kind of paradigm might change the way you and others in the profession practice medicine? Are medical schools talking about newborn screening now? Is it going to be a big part of the curriculum going forward? Because it's strikes me as something that is going to dramatically impact a small percentage of families that get an actionable finding, but then there's, double-digit percentage of families that get something that's somewhere in the messy middle, right, where they need to work it out with their
Yeah,
doctor and what the impact is.
yeah,
So what do you think how is that gonna play out?
yeah. Yeah, that's such a good question. So I can't tell you exactly what the medical school curriculum looks like, but I seriously doubt there's a big emphasis on newborn screening. I mean, I'd probably discuss it, at some point. And actually, just talking to the residency training program leads here at Children's about kind of genetics. It seems that medical students are coming into residency not necessarily with that great of a understanding of genetics. They probably understand the basics of genetics, but in terms of the interaction and the impact on clinical care is something that they don't really, I think, have much. It's hard to learn that in medical school, and even as residency, it's as time goes on, I think they probably learn more about that from doing it. But it's a very good question about kind of educating providers. And so what we do have in our second BabySeq project was an opportunity for healthcare providers to get some basic training in genetics, implementing it into their practice if they found something. And we started off as that was kind of a part of all the babies that we enrolled were family had pediatricians who had kind of gone through this training, and we've kind of loosened that up a little bit, but we still have a number of physicians in the practices from which we're enrolling who've gone through this educational piece. And this is something that Hudson Alpha down in Alabama has developed this very nice genetics education program for primary care physicians. And so I think as time goes on, it may be that will need to be more kind of education of providers about how to respond to this information. On the other hand, currently in newborn screening, if a pediatrician gets a result back, they send them to metabolism or endocrinology or wherever they need to go. So they kind of know enough to know that they're not going to treat this and they're going to refer. And so whether as this eventually kind of gets, sequencing gets integrated into newborn screening, there are more results come back. It's not really clear, I think, at this point, what's going to happen to those babies who have these findings. And my guess is that there'll be more kind of referrals to geneticists or cardiologists or whatever other providers to kind of manage. And that's what happened in the initial BabySeq project. So we had some we had some cardiomyopathies, we had hearing loss, we had a variety of things. And for those, when we turned those results, we recommended kind of suggested follow-up. And I think most of the families went and had follow-up and had whatever studies they need, including kind of cascade testing of other family members if needed, or an evaluation of a family member who is also carrying the variant. So it's a very good question. How is this all going to get integrated into primary care? And I think we don't really know at this point, but there probably going to need to be some changes in how that's done, because it's going to get more complicated.
Right, definitely. I was curious actually to talk about SIDS. So it's another big area that you work on that's in the same, realm. For those who aren't familiar with the acronym, it stands for Sudden Infant Death Syndrome. It's something that many or most parents are probably aware of, and frankly, it's a lot of parents' worst nightmare to even think about.
Yeah.
And at least from my perspective, it's not something I know very much from a scientific perspective. I'm a father. I have a 20-month-old. I worry about the temperature in her room and some of the things that they tell you to worry about. But there's a lot that we still don't understand. And you're leading the genetics portion, I think, of a major SIDS research program. So I'd love to hear a little bit more about that. What's the current state of what we know? what are some of the hypotheses, what are some of the hypotheses that you all are working on in the context of SIDS?
Yeah, so that's a great question. Yeah, that's a big kind of part of, also part of what I do. So I'm part of something at Boston Children's called Roberts Program for Sudden Unexpected Death in Pediatrics, which we call SUDP. So this program really spans both SIDS as well as sudden death in childhood, sudden unexpected death in childhood, which is SUDC. It's like a lot of acronyms, but and so these are, and the SUDC is kind of similar to SIDS. We have a child who just dies suddenly and unexpected, and trying to understand the cause. And so, so Rick Goldstein is the director of this program and the one who really started it. He's trained in, was a general pediatrician and then palliative care for a number of years and has done a lot of work in SIDS. And so I think, we see, and so I also say there's also a very important part of SIDS in terms of the Black to Sleep program and some of the epidemiology and the relationship between alcohol and smoking and, how the children sleep position. And so that's an important part of SIDS. And even, with the Back to Sleep program, there's still, that did lead to a decrease in sudden and SIDS. But there still is, a lot of children who die of SIDS. And those, particularly the Back to Sleep, is really just relevant in really younger children. I mean, once kids get to be, certainly by a year, and I would probably by 6 months, it's, the sleep position isn't as much of an issue because kids can sit up. But and they also, they're walking around, they have control. They're not, they're unlikely to kind of not be able to lift their head up. And then there's also the children who die over a year of age, where those are, it's really not, the Back to Sleep doesn't really come into it. And so then it's really, why are these children dying suddenly and unexpectedly? And so our kind of overall hypothesis is that SIDS and SUDC are really heterogeneous, are very heterogeneous. So there may be some children who have, just truly have suffocated. There may be some children that something happened. They might have had a seizure that wasn't appreciated, then that led to them being in the position and suffocating, or there may be a arrhythmia, or there may be some other condition that had not been identified that's responsible for them having died unexpectedly. And so we're taking kind of this genetic approach to understand and identify kind of underlying genetic associations with SIDS. And there are a number of programs around the world really looking at kind of in genetics and SIDS. I think what is unique about Robert's program is, first of all, I think we're like the largest program in terms of the number of cases that we've enrolled. But we also take a much broader approach. So we're not just doing genetics on the child who died. We're rolling families. We're doing actually trios. So we do genetics on the parents and the child to be able to identify what's de novo, what's been inherited. We do phenotyping. So we collect slides from the medical examiner's office. We look at the neuropathology as well as the general pathology. We collect family history data. We collect medical record data. We do a whole intake with the families. And so the families enrolled, they're just a big part of this program. And also, so Rick is as I said, he's in palliative care and has done a lot of research in grief. And so we also are then studying the kind of grief outcomes. What is it like to parent a child after you've had a child that had SIDS, and, and how do parents handle grief? So it's a program that's just very comprehensive, that's very family focused. And then so we not only enroll them, that we then report back to families when we're done about the things that we've found. And so we published a paper a couple of years ago in Genetics and Medicine, just, showing some of the variants, kind of the pathogenic variants that we were finding. And our kind of phenotypic hypotheses are really focused on epilepsy, cardiac conditions, particularly arrhythmias, kind of metabolic conditions. And then we've had a couple of children who have been eventually diagnosed as having a syndrome, a genetic syndrome that may have been then, that may have kind of led to a little bit of phenotype expansion of that syndrome, that sudden death could be kind of part of it. So, so that's really kind of what this program is about, and how we're viewing sudden death in childhood.
Yeah, and I guess a lot of this ties back to the overarching newborn screening, right? For everything that you discover here,
Right,
it's almost certainly an
right,
argument to include it in an upstream panel, so you
exactly,
could do something. You may not be able to, but
exactly.
at least you could try. Yeah.
Exact, exactly. I mean, that really ties those two projects together because if you could identify, and that's really, what part of what, really a driving force for this research is, can we identify children ahead of time who are at risk that can be intervened on to prevent this from happening, to kind of get them through this period, and then they go on and presumably, kind of lead healthy lives. So that is that intersection, and that is really our hope and our in the future to proactively identify children at risk.
Yeah, it's very important work. If we take BabySeq 2, and let's just assume for a moment that you find the same general themes of BabySeq 1, that parents don't have a major negative impact from the kind of screening that you do. And so this gives us comfort that this kind of approach is at best doing no harm. And then also, I think you've made a coherent argument that it's doing a lot of good. What next? What comes from there? And what is in the way of having universal newborn screening? If we get comfortable with the fact that we're not causing harm to parents and there's a lot of gain to be had, what's the next problem that we need to solve?
Yeah, I mean, the next problem is for us, I mean, I should say in terms of BabySeq, once we finish this randomized trial, we'd love to do a larger trial of just enrolling children and really looking to see what we found in the larger. And then you can, if you had, thousands of babies who were enrolled, you could really start to look at outcomes in terms of medical outcomes, healthcare cost outcomes, those type of things, which are going to be long-term outcomes. So within the context of a 5-year grant, we're not going to get as many of those. It's really, you need to have something that goes out for a number of years. So ideally, we'd have kind of eventually have more data. I think the issue with incorporating genetics in newborn screening is really a public health issue. And so I think it has to be, there's different ways you could do it. It could be something that was the people could do as an add-on if they wanted to. It could be more kind of voluntary in terms of that, or you could really build it into newborn screening. So there's different ways to do that. I think the problem is that newborn screening is regulated on a state-by-state level. And right now, to add one condition on the newborn screen takes the years. It has to go through a whole study group, that evaluates it, and they do natural history studies, and it's a whole process to get even just one condition. And so if we wanted to, newborn screening was to be a public health initiative, a sequencing, a public health initiative in newborn screening, I think that whole paradigm would have to change. And it seems like at this point, a pretty heavy lift. I'm not sure how that exactly is going to happen, but I think that's really what the issue is. Certainly in the United States, I think if you talk to, the people who are doing genomics, England, it's a little bit different. They have a national healthcare system. So their healthcare system is it's all tied together. So they can do a study that involves the entire country and may have more of an ability to implement something in the country. It's just ours is a totally disseminated with
Yeah.
each state making decisions, which and makes it hard to do. There's also things like cost, how long it takes to do the analysis. And my guess is that if sequencing were to add a newborn screen, it probably would be a pretty limited list in the beginning. The advantage is that if you do the sequencing, even if you return just a limited number of things, you still have all that data. So if you decided to expand it, you could, return more things as time went on, or add things it would be easy to add things because you'd already be collecting the data. It's just a matter of what you were returning. So
Right.
that,
That's
to
right.
me, that's a big issue, is how you're going to implement this on a public health basis, if and how it would be implemented in a public health basis.
Yeah, so I suppose you've created a model with BabySeq that other states or centers could adopt, right? And there's a potential that it could pick up on a state by state basis, but eventually someone's going to have to make the case. It sounds like on a federal level to say, hey, we've done a lot
Right.
of this work, big, well-controlled studies, and now it's time to make it accessible and uniform, right?
And that's what happens now with newborn screening. So there's a central, kind of a central national kind of committee that makes recommendations in terms of what should be in the newborn screening panel. And all the states have the newborn screening program. And so they have this RUS recommended uniform screening panel. And there's like 34 recommended, and there's like a certain number of 26 or something secondary conditions. And then states decide which one of those are going to put on their screen. So I think it would have to be at a federal level, the recommendation of sequencing and how the samples are collected, where it's done and what you return. And then states would decide. I mean, if we kept the same paradigm, which I think we would, states would then decide what they wanted to return.
Yeah.
I mean, the problem with this is that it then means that what you get back depends on what state you live in, which is still kind of the problem with newborn screening, and potentially would be exacerbated by adding this, because some states might say we can't afford to do it. And something it's just a lot of moving parts.
Yeah. How are you all addressing that? Can you address that affordability or the health economics question in BabySeq too, or is it not big enough or long running enough to answer that question? If we spend a few thousand up front on this testing, counseling, and return of results, does or doesn't that pay off over a child's lifetime?
Yeah. That's such a good question. I think you would need really large cohorts of individuals enrolled to start to see how many diagnoses that you're making that are going to change healthcare costs. So because it is not just the healthcare costs, you said it's the cost of the sequencing, interpretation. And I think there are people who are kind of certainly in our group, we have a group that's the Harvard Pilgrim that has kind of our economic analysis that's led by Kurt Christensen and some people working for him, but kind of trying to look at the healthcare costs. And eventually that is the idea. But I think you would really need, thousands of babies enrolled in these studies. And it may be that all these studies are be doing kind of in different places, like the Guardian or the UNC, if we all got together and try to do some follow-up. But I think that is an area that I think the newborn screening community who's looking at sequencing newborn screening, is very interested in how we could kind of start to answer that question. But it's going to take I think a lot of babies enrolled.
Yeah,
to really look at yeah.
Yeah, I agree. The data pooling idea is a really good one. And I suppose you could couple that with, there's enormous data sets of what happens if you don't do this, right? So you have the whole healthcare system is basically what it costs if you don't do this, right? And then if you can put together enough cases, you have the advantage in BabySeq that you have the randomized controlled trial, where other ones you're going to have some ascertainment bias, right, that make it a little bit different. Right. But maybe smart statisticians can crack that problem.
Yeah. I should just also kind of make a plug. There's an organization called ICONS, which is an international kind of organization for looking at newborn sequencing. And so they've started, I think, a few years ago. And so all of these studies are kind of part of ICONS, and they have this kind of meeting once a year and calls kind of once a month. But that's a kind of thing that ICONS is really thinking about, is that how can we think about implement, all these studies, what we're learning, and implementation. And so these are the types of things I think that they're also kind of talking about. Yeah, they had a meeting recently in the fall in London at the Royal Institute.
Yes, that's right.
In London. So
it's a great plug. And for those who are interested, the website is iconseq.org. And the next one is going to be in New York in 2024, October 9th through 10th. So if you're interested in this kind of thing and you want to meet everyone who's working on it, I think that's the place to go.
Yeah.
Just to close up here, I'm wondering, okay, I'm going to ask you to do more predicting the future. Let's suppose we work out the questions about do no harm. So we're comfortable that we've structured a system that does no harm. We figured out a way to pay for it where the numbers check out. We can spend this kind of money up front and we get good outcomes. How does that change pediatric healthcare? What's different if we suppose we're a decade or two from now and every child in America goes through the kind of program that you've set up at BabySeq? What's different about pediatric healthcare, for better or for worse? Hopefully, mostly for better. But I'm interested in your perspective.
Yeah, I mean, I would hope that pediatric healthcare would be better. that children who are at risk for things like, well, if you take retinoblastoma. So there's a group here very interested in Lisa Diller and Rich Perrod at the Farber, and the children's are very interested in retinoblastoma. And, kind of if we were then to be able to do genomic sequencing, we identify someone with a risk for retinoblastoma, it potentially prevent cancer. So retinoblastoma is a cancer in the eye, develops in young children, and you could, have eye exams and do whatever you needed in terms of, taking out the eye if you had to, whatever, to increase. So these children who would have otherwise passed away would not, because they would get treatment. So those are kind of, I think, some kind of clear things that we really potentially can prevent diseases in some people. We can treat people earlier with kids earlier, and so that they do better. And even things, if there were things on there that weren't necessarily treatable with the medication or whatever, we could also intervene earlier, help parents understand what's going on, help them, preventive, kind of predictive, what's future issues to deal with sooner. So I think there's a lot of kind of opportunities for improving pediatric healthcare by developing and treating diseases earlier. And the other thing to say, even these conditions that don't seem to be treatable at this point are becoming treatable. So there's, gene therapies are kind of coming up that may be treatable. And then there's this whole area of, kind of N of 1 therapies. So, allele specific oligonucleotides, for example, and other ways of targeting variants. And if you have a child even who has a condition that may in the long run be devastating from a neurologic perspective, if you knew about it early on, you could go in and do some type of a therapy to, CRISPR or this ASOs or gene therapy to really change it. You potentially could change the course of even these things that we don't think are treatable right now.
Yes.
So as time goes on, they may become treatable. And so, that's probably the most exciting kind of implication for these, is that you may have children that you are going to prevent kind of devastating neurologic diseases for things where they're kind of fine at birth but develop. I mean, there'll be some children, obviously, things are born with that we're not going to intervene, but there are a lot of conditions that are progressive with time, and that may be that we can intervene on a genetic level in 10 years.
Yes.
So that's probably one of the most exciting aspects of this kind of sequencing and newborn screening.
I couldn't agree more. I was at a major genetics or a major conference for ALS, also known as motor neuron disease in the UK. And there was a young girl on stage who had received one of these ASO therapies for her FUS mutation, which is a ALS that's that primarily happens in young children, and it's very aggressive and fast progressing. And you've got to wonder, she was in her teens, and she displayed actually a pretty dramatic recovery as a result of this ASO. She went from not being able to walk to being able to walk again. And you've got to imagine how the world could be different if that gene was picked up when she was newborn rather than starting clinical symptoms when she's 12, right? And you times that by 7,000 rare diseases, you've got something amazing.
Right. And we think all of these, these kind of diseases are not treatable, but now that's the whole paradigm, is that they are becoming treatable.
Yeah.
So, so that's probably the most exciting part of this, is that, combined with therapies will be available with newborn screening, and what a difference we can make in people's lives.
Great. A great and very optimistic note to end on. Thank you for your time today and for spending so much of your career on these important things. It's obviously a big difference, and I think the best is yet to come, hopefully in the next couple of years.
Yep. Thank you so much for having me.
My pleasure. And thanks everyone, as always, for listening. If you like this episode, the thing that we would really appreciate is if you just share it with a friend or colleague that you think would like the episode too. Thanks again, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Sanogenetics on Twitter, LinkedIn, or visit sanogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.